Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to our channel. We are here with another video. Today our topic to discuss is viral evolution, morphology and its classification. Basically we are going to discuss virus. So let's take a start. Viruses are infectious particles about 100 times smaller than bacteria and can only be observed by electron microscopy. Learning objective learning objective describe how viruses were first discovered and how they are detected. Here are some key points. Variant single virus particles are 20 to 250 nanometer in diameter. In the past, viruses were classified by the type of nucleic acid they contained, DNA or RNA, and whether they had single or double standard nucleic acid. In the last point of key point is molecular analysis of viral replicative cycle is now more routinely used to classify viruses. Key terms vary on a single individual particle of a virus, the viral equivalent of a cell. Now talk about virus. Discovery and detection. Discovery and detection. Viruses were first discovered after the development of a porcelain filter called the Chamberlain-Pasteur filter, which could remove all bacteria visible in the microscope from any liquid sample. In 1886, Adolf Meyer demonstrated that a disease of a tobacco plant's tobacco mosaic disease could be transferred from a diseased plant to a healthy one via liquid plant extract in 1892. Dmitry Ivanovsky showed that this disease could be transmitted in the way even after the Chamberlain Pasteur filter had removed all visible bacteria from the extract. Still, it was many years before it was proven that these filterable infectious agents were not simply very small bacteria but were a new type of tiny disease causing particle. Here is the diagram of mosaic virus. This picture is about mosaic virus. Mosaic virus, the structure of the mosaic virus. In the past, viruses were classified by the type of nucleic acid they contained, DNA or RNA, and whether they had single or double standard nucleic acid. Virions single virus particles are very small, about 20 to 250 nanometer in diameter. These individual virus particles are the infections form of a virus outside the host cell. Unlike bacteria, which are about 100 times larger, we cannot see virus with a light microscope, with the exception of some large version of the pox virus family. It was not until the development of the electron microscope in the late 1930s that scientists got their first good view of the structure of the tobacco mosaic virus TMV and other viruses. The surface structure of virions can be observed by both scanning and transmission electron microscopy, whereas the internal structure of the virus can only be observed in image from a transmission electron microscope. The use of these technologies has enabled the discovery of many viruses of all type of living organisms. They were initially grouped by shared morphology. Later, groups of viruses were classified by the type of nucleic acid they contained, DNA or RNA, and whether their nucleic acid was single or double standard. More recently, molecular analysis of viral replicative cycles has further refined their classifications. These are the image, these two image taken by uh, electronic microscope. This is the view from microscope taken.